So now, welcome back to BioMindscape. Let's solve botany questions of Neet 2026. So which one of the following is triploid cell? Synergid, zygote, central cell and primary endosperm cell. So here the correct answer is primary endosperm cell. Then which type of pollination bring genetically different type of pollen grain to the stigma? Autogamy, genetogamy, xenogamy and cleistogamy. So here we know that there are different type of pollen grain. This is xenogamy. Then next question is match the placenta type like marginal, axial placenta, then parietal and basal. We have to match the list 1 with the list 2. So here list 2 is mustard, pea, marigold and lemon. Let's see the option 1. A. Marginal placentation. It is matching with P. Correct. Axial placentation. That is matching with which one? Mustard, marigold or lemon? Lemon. So we can directly get the answer, that is option 1 here, because option 1 is correct. Parietal placentation, it is showing mustard, and D, that is B cell placentation, we can see in the marigold. So here the question is, in the 5 kingdom classification of R.H. Whitaker, which one of the following is not a main criteria? So here the cell structure, B body organization, C is phylogenetic relationship and D is the locomotion. So here the correct option is locomotion, because other than that the cell structure, body organization and phylogenetic relationship, all these other are the 5 kingdom classification. They are the main criteria. Next question is the reaction. C single bond C, carbon single bond carbon, carbon double bond carbon plus X and Y. It remove the leading to the double bond. Option is transferase. It is used in transfer, so this is not the correct option. Hydrolase, it is also not the correct option. Lyase is the correct option and not the ligase. Now why, we will know. Lyase catalyze the removal of the group from the substrate by a mechanism other than hydrolysis that often result in the formation of a double bond. So here example is dehydrogenase. So dehydrogenase and deaminase. Whereas transferase, it is used in the transferring of the group, and hydrolase, it is used in water, hydrolysis of water. You might have seen this, hydrolysis of water. Then next is lyase, that is joining of the molecules. We can use in joining the molecules, ligase, whereas the correct option for this is lyase. Next question is, which one of the following bioactive molecule is obtained from the plant and used as alkaloid? Here let's see the other option. What are the options here? Option 1 is trypsin, option B is collagen, option C is morphin and option D is concavenyl concavenyl A. So here the correct answer for this question is the morphin. Morphin is the correct answer. Now let's see why morphin is the correct answer. You know what? Morphin is alkaloid that is obtained from papaver somniferum, that is Opimum poppy, whereas trypsin, it is an enzyme in a protein. Whereas if we see the collagen, what is collagen? Collagen is a protein that is present in intercellular group substance, intercellular grounded substance in the protein. Valine A, it is a lactin that is formed from the jack beans. Concavenyl A, that is forming from the lactin. Now type of placentation in pea, that is marginal placentation, we can see that is formed in a pea plant. In pea, that is family Fabaceae. Family Fabaceae, what are the things that are like ovaries monocarpellary in family Fabaceae? They are arranged in a ventral pseudo, that is marginal placentation. Which one of the following is correct regarding embryonic site development in angiosperm? We have seen that is primary endosperm cell in the triploid. So why is it after triploid fusion? What happens after the triploid fusion? The primary endosperm nuclei pen, that is in 3n, that is triploid, and it is divided to form the endosperm tissue which nourishes the development of the embryo. Whereas synergids, they are haploid, central cell is diploid before fusion, and zygote is diploid. So here are some questions. Which one of the following is not an example of convergent evolution? Option is flippers of penguin and dolphins. Option B is eye of octopus and mammal. Option C is forelimbs of whale and bats, whereas fourth option is wings of butterfly and birds. So here the correct option is forelimbs of whales and bats. Next question is, male frog can be distinguished from the female frog due to presence of option A bulging eyes only, option B vocal sac and coupling pad on the forelimb. So this is the correct answer, B, vocal sac and coupling pad on the forelimb. Option C is olive green skin with dark spot. No, because both have, male and female frog both have the olive green color skin with the dark spots, whereas the webbed digits in feet both have the similar characters. All others are the similar characters, whereas the vocal sac and coupling pad on the forelimb, this is the different one. Male frog can be distinguished from the female frog due to presence of the vocal sac and the cupulatory pads on the forelimb. Next question is, streptokinase is used as option A blood cholesterol lowering the agent, B immunosuppressive agent, option C is detergent formulation, and D is the clot burst, that is removal of the blood clot. So here the streptokinase, it is used in the removal of the blood clotting, that is the clot burst. Next question is, so here the question is, the juxtaglomerulus apparatus is formed by the cellular modification of option A proximal convoluted tubule and Henle's loop, option B is distal convoluted tubule and afferent atriole. So this is the correct option, distal convoluted tubule and afferent atriole, whereas C is collecting duct and afferent atriole. This is wrong. Bowman's capsule and PCT, this is also wrong. So the correct option is option B. Now moving on to the next question, which one of the following is incorrect regarding the Rh grouping? Rh antigen is present on the RBCs of most human. This is true. Erythroblast fetus occurs when mother is Rh negative and fetus is Rh positive. So here the option B is incorrect statement. It is reverse given. Then option C, anti-Rh antibodies are given to the mother immediately after the birth of the first child. Yeah, Rh incompatibility is mainly observed in the subsequent pregnancy. So here we have to tell the incorrect option. So option B is incorrect. Now moving on to the next question, volume of air in the remaining of the lungs. The volume of air that remains in the lungs after a normal expiration is called the tidal volume, or inspiratory reserve volume, or expiratory reserve volume, or residual volume. Tell me the answer. So the answer is residual volume, because it is the volume of air that remains in the lungs after we normally expire. You know, inspiration and expiration. Expiration means we are taking in breath and taking out the breath. So taking out the breath is expiration. So the amount of air that is remaining in the lungs after normal taking out of the breath, that is expiration. That is the residual volume, that is remaining one. Then what is the normal value of tidal volume in a healthy adult human? That is 500 ml. Now I have made a video on this, all the respiratory volumes. You can go and watch on BioMindscape. Now moving on to the next question. In the person with the O blood group, the genotype is I, small I, because all other when they are present, capital I, capital A and capital A, when there is A blood group, capital B when there is B blood group, AB when AB blood group is present. So the correct answer is D. Now probability of the child having the blood group O when both the parents and heterozygote, they are given here. So the answer for this is 25%. So here the question is, which one of the following bioactive molecule is used in immunosuppressive agent? So here the option is statin, option B is streptokinase, option C is cyclosporin A, and option D is lipase. So here the answer is cyclosporin A. The next question is, the enzyme that help in the removal of blood clot is the streptokinase. So here the match the pair is given. We have to solve about 65 million years ago, about 500 million years ago, about 350 million years ago, and about 320 million years ago. And list 2 is given. Jawless fish probably evolved. Option 2 is the dinosaurs suddenly disappear from the earth. Then seaweeds and few plants probably exist. And option 4 is invertebrates were formed and become active. So we have to find out the correct option. So here we can see the option 1, A is 3, A is 1 into A is 2 and A is 2. So we can eliminate all those because 3 and 4 are the option that is showing A is 2, that is about 65 million years ago, that is the dinosaur suddenly disappeared from the earth. Now let's see the other option. About 500 million years ago, invertebrates were formed and become active, that is Cambrian explosion period. Option C, that is about 350 million years ago, the jawless fish probably evolved. And option D is about 320 million years ago, the seaweeds and the few plants probably exist. So this is the correct option, option 3. Now moving on to the next question. The WBC count of a person's blood sample is 8,000 per QMM. How many eosinophil and lymphocytes would be in the same blood sample respectively? So the option 1 is 300 to 500 and 1200 to 1500, whereas 160 to 240 is the answer. So normal WBC count is 4000 to 11,000. Here it is 8,000. Eosinophil is 2 to 4%. So this question, it is coming from the blood part of your NCRT. Directly this question is asked. The amount of lymphocyte at 20 to 40% of WBCs, that is 1600 to 3200, typically 20 to 25% ranging given as 1600 to 2000, that is 1600 to 2000 in the option. So here we can see the correct answer is option 2. So this is from the human physiology. Here the human protein name alpha 1 antitrypsin obtained from the transgenic animal is used in the treatment of option A, epenthema is the correct option. So here the question is, the following are the stages of the life cycle of the plasmodium. We have to arrange the stages in a correct order. So here the first option is correct one, because first the female mosquito inject the sporozoites into the human during the bite. After that the sporozoite reaches the liver through the blood, and then the parasite reproduce asexually in the liver cell, bursting the cell and releasing into the blood. And after that the parasite reproduce asexually in the RBC, bursting the cell, and then gametocyte develop in the RBCs. So this is the correct answer. Here we have to arrange the renin and antagonistic mechanism in a correct order. So here we can see there is increase in the blood pressure and glomerulus filtration rate, reabsorption of Na positive and water from the distal part of the tubule, then fall in the glomerulus filtrate rate and vasoconstriction by angiotensin 2 and release of the aldosterone, renin converting into the tensinogen into the angiotensin 1 following by the angiotensin 2. So we have to arrange them in a correct order. So your correct order is fall in the glomerulus rate. After that, renin will be converting into the angiotensinogen into the angiotensinogen followed by the angiotensin 2. After what happened, this angiotensin 1 will be renin is getting converting the angiotensinogen into angiotensin 1, and that is followed by the 2-1. After that, the vasoconstriction of angiotensin 2, and then there is release of the aldosterol. Okay, aldosterol will be released, and after the release of the aldosterol, what is happening? The reabsorption of the sodium. The sodium will be reabsorbed and water from the distal part of the tubule. The what is happening? First, the sodium is getting reabsorbed, and after that, the water that is getting absorbed from the distal part of the tubule. After that, the next point is the increasing in the blood pressure and glomerulus filtration rate. So the correct order for this is C, E, D, B, A. This is the correct sequence. So correct answer is this one. Now moving on to the next question. So here this question is we have to arrange the steps of respiration in human in the correct sequential order. First is diffusion of O2 and CO2 between the blood and the tissues, differentiation of the O2 and CO2 across the alveolar membrane, pulmonary ventilations, breathing in and out. D is cellular respiration, and E is transport of the gases by the blood. So here the correct option, sorry, sequence is first pulmonary ventilation, that is breathing in and breathing out. After that, the diffusion of O2 and CO2 across the alveolar membrane, and after that, the transport of gases by the blood, and after the transport of gases by the blood, the diffusion of O2 and CO2 between the blood and tissue, because after the gas is getting transferred by the blood, there is a diffusion that is occurring between the O2 and CO2, blood and transfusion, and after that the cellular respiration occurs. So this is the process, the step, that is C, B, then E, A and D. So this is the correct sequence. So here you have to match the following. First list one is the cortisol, aldosterone, cholecytosine, progesterone, and list 2 is given stimulate formation of the alveoli in the mammalian gland, production of anti-inflammatory reaction, stimulate reabsorption of sodium in water from the renal tube, and stimulate secretion of pancreatic enzyme and bile juice. So here the correct option is A, that is cortisol. It is pairing with the products anti-inflammatory reaction. Then aldosterone is pairing with the stimulation of reabsorption of sodium and water from the renal tube. Then cholecytokinin. This cholecytokinin, it stimulates the secretion of pancreatic enzyme and bile juice, and progesterone and stimulate the formation of alveoli in the mammalian gland. So this is the correct option, A2, B3, C4 and D1. So here the question is insertion of the foreign DNA and BAMH1 site is in the E. colonial vector PBR322 result in the loss of antibody resistance towards option 1 ampicillin and tetracycline, option 2 ampicillin, option 3 tetracycline, and option 4 gentamicin. So here the correct answer for this question is tetracycline. Why tetracycline is the correct answer? Tetracycline in the plasmid PBR322, the BAMH1 restriction site is located inside the tetracycline resistance gene. When the foreign DNA is inserted in the BAMH1 site, it disrupted the tetracycline resistance, and the ampicillin resistance gene remain intact, and therefore the recombinant bacteria will be resistant to ampicillin but sensitive to the tetracycline. So this is classical selectable marker example used in the cloning.